Good afternoon, everybody. My name is Rachel Caston. I'm the Student Services Specialist here in the Center for Geospatial Analytics, and I have the immense pleasure of working very closely with our Ph.D. and Geospatial Analytics students, and you are going to hear from a few of them today. So we're going to start with Eli Horner, who is a fourth-year PhD candidate here at CGA, and his research focuses on uncertainty analysis for spatial models, specifically quantifying and partitioning uncertainty. As an undergraduate, Eli majored in applied math. Outside of work, he loves spending time outdoors, at least when there is less pollen than there is right now. So give it up for Eli.
Hi, everyone. It's over here, not on me. So I'm going to talk today about my research on partitioning uncertainty in spatial forecasts, and specifically looking at ecological forecasts, because there's so many different things that we can forecast ecologically, from invasive species to phenology to water quality. And by forecasting this, we can help understand potential future conditions and plan management strategies. So for example, here, like in a scenario where we're thinking about how many trees might become infected with or without treatment. But in my lab, we try to focus not just on forecasting, but specifically spatial forecasting, where we get to add in this layer of where, in addition to the when and how much that we're kind of getting from this traditional forecasting. And we also like to focus on iterative forecasting, where models are updated with new data as it becomes available, because this is going to help improve our forecast accuracy. Whenever we think about modeling and forecasting, though, we need to think about uncertainty, because sources of uncertainty and their effects must be quantified to avoid making falsely overconfident predictions. We also have some practical management benefits here. So if we imagine a scenario where we're looking at two models to predict something, and they both seem to be saying the same thing, but then we add in uncertainty, and they are actually saying different things. So in one of these cases, we're pretty confident in our prediction, whereas in the other one, there's a lot of variation. And if you have limited resources to spend, these might mean spending a lot of money on something or spending very little because you really don't know exactly what's going on. Once we quantify uncertainty, we can move on to this next step of partitioning uncertainty, where we try to break down our uncertainty into its different sources and say how much is coming from each source. And in ecological forecasting, we consider four main sources of uncertainty. We've got driver uncertainty, which comes from the unpredictability or limited knowledge of the future state of terms called drivers. They're kind of affecting our model's forecast, but aren't being directly forecasted by the model. We've got initial condition uncertainty, which comes from the fact that the starting state or the current state of the system is not really perfectly known. We've got parameter uncertainty, which comes from how well our model parameters are describing these real-world values that we're calibrating them to. And we've got process uncertainty, which is coming from unexplained variability not captured by the model. So when we put these together and we kind of break them out from our uncertainty, we can get diagrams that look kind of like this one on top, where we're looking at a forecast and we're saying, of its uncertainty, how much can be assigned to each of four sources over time. And what you'll notice is you can see different points in time where some sources are more or less important than the other sources. But what we can also do is we can go beyond that and we can add this spatial component to this. So this is a bit of like a sneak preview of some figures I'll show later. But here, each of these little cells or pixels is colored by its largest source of uncertainty at one point in time. But we don't just have that. We can actually imagine each of these little cells has one of these inside of it. So just the amount of information that we can gain from doing this is massive. Like we get so many of these little graphs, just in this one image. So this is really cool. But unfortunately, spatial uncertainty partitioning is not currently being done. In fact, uncertainty partitioning of any kind is really rare. So my lab group did a review study where we looked at over 400 papers, and we found that only about 20% of them even report uncertainty at all, let alone partition it. So among even that small 20% that report it, only about 8% of those go on to partition it. And that's where my work comes in. I developed a tool to help perform spatial uncertainty partitioning using something called the Sobol method. We're going to demonstrate this tool here with a case study, and it's going to tell us about both when and where the uncertainty is high in our study system. And the study system we're going to focus on is sudden oak death, which is a forest disease caused by the invasive pathogen Phytophthora ramorum. It infects and kills trees on the West Coast, which lead to some significant ecological and economic impacts in the area. We see over here we've got a map of different counties where we have confirmed sudden oak death infections. It was first discovered in the Bay Area and has since spread northwards. California, it's been relatively unchecked, the spread, but in Oregon, they've invested a lot in managing sudden oak death, and that's why for this study, we're going to focus on Curry County as our study region in Oregon, where we are working with the Oregon Department of Forestry. They've set up a quarantine area in Curry County down here in our study area, and one of the main goals here is to contain the spread to the quarantine area and specifically prevent spread to Coos Bay, which is a major port up here, very important for the timber industry, which is really the main industry in this area. If the infection were to spread there, that could lead to port closure, and that would be a really negative thing. We really don't want that to happen. And so we're going to use our spatial uncertainty partitioning tool, and we're going to use this to help tell the Oregon Department of Forestry what the most efficient way to collect data is to help them support these management goals. So to do that, to analyze our uncertainty in our model, we need to model. And what we're going to use for our model is the POPS model. And the POPS model simulates the spread of sudden oak death using 3 different processes. So we look at reproduction, dispersal, and establishment that we're simulating. And here, each of these terms in this equation are color-coded by the source of uncertainty that they contained from earlier. And what I want to point out here is mainly that our weather uncertainty is really high. Because we're predicting out 5 years, and 5 years of forecasting weather is an impossible task. However, because it's so high, we're actually going to show results without this, because what we're really interested in is this idea of targeting data collection and management impacts, which we can't really, it's not really helpful to say, you don't know what the weather is going to be in 5 years, and that's a problem, because they can't do anything about that. But they can do things about the other 4. So that's what we'll focus on. So I'm just going to take a look here in our study area in Curry County, some sudden oak death infections. So these are our predictions as we move out 5 years in time. We can see the infection growing and spreading in both size and direction. And what I want to kind of focus on here with this simulation is these 3 different zones of infection that we can think of. And they're not exactly like hard and fast locations. As we move through time, they're going to spread as the infection spreads. But we've got 3 main zones that we think of. We think of this epicenter zone, where we have the most infections. We think of this wave front, or this kind of leading edge. So as the infection's spreading out, that leading edge moves out, that epicenter moves out. And then finally, we think of this long distance zone, where we don't have a lot of infections, but especially for things like quarantine and preventing spread to certain places, these long distance infections can be really important. So it's important that we capture what's going on there as well. We're going to come back to this a little bit later when we think about how uncertainty might be different in these 3 zones. But to talk about uncertainty, we need to talk about our method for partitioning uncertainty. So we use something called the Sobol method, which allows us to create this thing called a Sobol total order index. I've got the formula down here, but I'm going to walk through it with these fun little colored boxes instead. So each of our columns here is representing a different model run, and each row is representing a different source of uncertainty. So for each model run, if the box is colored in, what that means is that we're including that source of uncertainty in the run. And if it's not colored in, that means we're not including it. So for example, in this highlighted run we've got here, we're including initial condition and parameter uncertainty, but we're not including driver and process uncertainty. So if we want to, say, calculate the uncertainty contribution of process uncertainty, what we first want to do is we want to think about, for the other 3 sources, what are scenarios where they are the same? So the only thing different between our scenarios is process on or is process off. So like this here, we've got all 3 on, all 3 on. The only difference is process on or off. So we're going to take the variance between those two outputs, and then we're going to repeat that for every single one where we've got this same kind of identical set for the other 3. So like here, we have an identical set. We take the variance, we take the variance. We're going to do this for all of these. And when we get done doing that, we'll take the mean of that variance. That's this part here. We've got the variance here, the mean here. And finally, we'll divide it by the variance of all of our runs put together. That's going to give us this total order index, or the percent uncertainty contribution of process uncertainty. So by doing that, we can get a traditional uncertainty partitioning diagram, which you might remember from earlier. So we can see the one on the left is showing that over time in our entire study area, as our prediction for total infections goes up over time, so does our uncertainty in that prediction. The one on the right here is what we want to focus on. This is our uncertainty contribution. And we can see that as we go through time, especially in the later years, our most important source of uncertainty here is host uncertainty. And so this is a useful result because we can go to, say, the Oregon Department of Forestry and say, when it comes to uncertainty and total infections in your study area, host uncertainty is most important. So we can tell them, if you want to collect data on something to make our model more certain and more accurate, host is the best bang for your buck. And this is great, but it doesn't account for the fact that the study area is very big. There's a lot of little areas. So just because we can say this for the whole study area, host is most important, doesn't mean that host is most important everywhere. And so what we can do is we can make these maps. And we can look at every single little cell, and we can say, in each year, what is the most important uncertainty source here? And we can do this, and we get this for year 1, 2, 3, 4, and 5. There's a lot going on. We're going to go back to our conceptual zones, right? We're going to think about the zones. So if we think about the epicenter zone, what I want you to do is kind of look in here in the epicenter zone. You'll notice you see a lot of green. We find that process uncertainty is the most important uncertainty source in the epicenter zone. In the wavefront, or the leading edge of infection, we'll want to focus in on trying to see that zone. What we see is that we see a lot of host, a lot of parameter, and some initial condition uncertainty. So we find those to be more important there. And then finally, in this long distance dispersal zone, there's a lot of these gray cells. And what these mean are that each source is contributing equally, or that there's no one dominant source in these areas. What's going on here is that these are areas where it's hard for an infection to spread, so we really need that interaction between all the sources for that to happen. And this is really cool that we have this conceptual framework. And one of the things that we want to do with future study is see, does this hold up for other species? Or is this just sudden oak death specific? But we also can look at things like a specific management example. So in this case, we've highlighted an area that's north of our epicenter zone, which if you remember from earlier, Coos Bay is up here. We want to prevent spread up there. So this might be an area that managers are concerned with. And what we want to do is we want to ask, if we do our analysis just in this subregion, what do we find? Is it the same as the whole study area, or is it something a little bit different? Is this spatial region a little bit different? Is something else going on there than going on if we consider everything? And what we find is that yes, there is something different going on. So while overall we found that host uncertainty was the most important contributing source, especially in later years, here we see something different. Host uncertainty is most important in the first year, but is becoming less important over time. Meanwhile, parameter uncertainty is becoming more important over time. So what we can go to them and say is we can say that if you're interested in spread specifically in this subregion, you're going to be better off collecting more data on the parameters of sudden oak death rather than where the hosts are. And we can also look at the maps again. And one thing that I think is really cool about this is that we kind of see a repeated version of the same pattern as before. So if we remember, we had that epicenter zone where process was dominant. This area down here is where we see the highest chance of infection in year 1, and we can kind of see process dominant here. This next ring out, we've got that same, we have the other sources of like host, parameter, initial conditions coming in, and then at the further distances, we've got a lot of these gray cells. And if we follow it forward 2 more years, we can kind of see the same thing. We've still got this process area right in here where there's the epicenter of infection. Even in this little like smaller area, we've got this like next ring out where we're seeing a lot of parameter and host. And then we've got this really far spread. We also will notice that there's a lot of spread between year 1 and year 3. And then even more spread when we go out to year 5. And you can start to see why parameter uncertainty might be dominant here, because you're starting to see these larger regions where we've got parameter uncertainty as the most important source. So to bring it all together, we developed a tool to perform spatial uncertainty partitioning in near-term iterative spatial forecasts, specifically shown here with a case study of sudden oak death in Oregon. We're planning to use this tool on some future species in the future. And finally, what I want to mention is that this tool is not just for the POPs model. We can actually use it for just about any spatial model as long as you can turn on and turn off your uncertainty. And the goal here is that hopefully this can be shared and used by other researchers so that we can get to a point where we can start to address that gap from earlier of, not a lot of spatial uncertainty partitioning being done. Thank you.
So I'm going to ask you to hold on to any questions that you might have. We're going to have all 3 of our speakers, and then we're going to do questions at the end. So make a mental note. We're going to swap some mics here. So our next speaker is Gwen Kershke. Gwen is a 3rd-year PhD student in the Youngstedt lab, and Elsa did a wonderful forum for us very recently. Hopefully you were there. Her research combines field ecology and geospatial analytics to address questions in pollination ecology.
Awesome. You folks hear me? Okay. Oh, forgot my clicker. Okay. Yeah, so I'm doing a slightly different format of talk here. Rather than being a results-focused talk, Esimi and John asked me to talk a little bit about the field campaign that I ran last summer and what that looked like. So my research interests long-term have been around bees and their floral resources. And so bees are getting 100% of their food resources from flowers in the form of nectar and pollen. And they're foraging for those resources in this kind of larger area that's around wherever their central home nesting location is. And the distribution of those floral resources throughout that space is important for understanding things about bee populations because it determines things like what their foraging efficiency can look like. But pretty much everything that we know about these floral resources is coming from discrete sampling points. So like this is me as an undergrad sampling floral nectar from this specific plant in this location. Also, most of the information that we get about floral abundance, so like how many flowers are out there, is also coming from kind of static sampling locations. So things like a plot or a transect that people are coming back to over and over, but they're counting like this is how many flowers are here, and we're assuming that the area around that is similar. And so my work in grad school has been focused on trying to bridge that kind of discrete information that we have about these floral resources with the kind of spatial scale that bees are actually encountering those resources at. And I had a really incredible opportunity to work on this last summer in collaboration with the Colorado Headwaters Ecological Spectroscopy Study, or CHESS, that was happening at the Rocky Mountain Biological Lab last summer. And so this overall project was a repetition and expansion upon a similar project that had been run in 2018. And for both these projects, they were primarily interested in things like some things about land cover change, leaf traits, hydrology, those kinds of questions. But as part of their project, they had contracted for the NSF NEON's aerial observation platform to come over this area around the Rocky Mountain Biolab. And that platform, among other things, captures hyperspectral imagery at a 1 meter pixel size with 426 spectral bands in the visible, near, and shortwave infrared. And so when I heard that this project was happening, I thought that this could be a really incredible opportunity to try to get some information on the ground simultaneously to that imagery that would about the floral resources that I could then combine with that imagery to try to do some of this scaling across space. And so kind of what did the process of that look like timeline-wise, right? I had learned about this project the previous summer, had a bunch of different planning meetings with the CHESS crew about what that might look like, tried to figure out what my project would be looking like. Then I spent the spring writing a bunch of grants to try to fund the project, as well as writing research proposals that I need to use to get the permissions to work there. Right? I'm doing this work mostly on national forest land. And so doing a lot of that in the spring, then coming into the actual fieldwork period in the summer. There was also a lot of coordination that was happening here between me and the CHESS crew and between them and the flight crew. So we're trying to get our field data, both myself and CHESS's field team, we're trying to get our field data as kind of concurrent with the flights as possible. And so we had to figure out like which flight lines are they flying today? What did they get done? Oh no, they had to go home early because it was too windy, which happened quite a few times. And so we're trying to then coordinate like, okay, we have to change our plan for what we're sampling on this given day in relation to that. Then there was in the fall, a bunch of data entry and cleaning of my field data that needed to happen on my end, as well as both NEON and a team at the NASA Jet Propulsion Laboratory have done a ton of pre-processing of the imagery. And that has just now been released. Last week, I was in Colorado at a workshop where we were just starting to get access to these data, which is very exciting. But that means that the kind of analysis, writing, and all of the reports that I have to write back to the grants and the permissions agencies are still upcoming. But what did the work actually look like on the ground? So these are the 8 field sites that I was working at. So these are distributed throughout that valley that I was showing earlier around the Rocky Mountain Biological Lab. And 3 of these sites were shared with the CHESS crew, and then the others were sites that were part of various different long-term either bee or flower monitoring projects, so that I then had access to other sets of floral resource and bee data that I can potentially use as well. And so the process of going out to actually do this sampling was, so we were interested first in getting information about the floral abundance that's there. So how many flowers are out there? That process was first to go around the site and flag out, like, where do we actually want to measure these flower numbers? And that was done by the kind of variables that we were considering there was trying to capture the variation in vegetation cover in the area, and then also trying to have the area around the plot be pretty similar to what's in the plot that can help us like deal a little bit with if there's issues between matching up the GPS coordinates exactly with the right pixel, that we're still getting pretty similar information. Then we would go out, actually put down our quadrat to say this is the meter we're looking at, count every flower of every species that's in that square meter, and then get our kind of high accuracy GPS coordinates of where that plot is so we can match it up with its pixel in the imagery. This is what that looks like. Well, this is what that looks like for a given site. So we had 60 quadrats per each of our 8 sites for a total of 480. We did this over a series of 5 days. They were 5 long days. Yeah, and then the other set of data that we were also getting at the same time was information about the nectar and pollen. So we know right from the first part the number of flowers, and we want to be able to scale the number of flowers by how much of each of these actual food resources that the bees are interacting with are in those flowers. And so on the pollen side, that meant collecting anthers in the field, which is what has the pollen on them in a flower, and bringing those back to the lab. And then there has been this awesome crew of undergraduate students who have been working with me in the lab this academic year to count all of those pollen grains, which has also been huge, huge amount of work. But we're getting there. We're getting very close. And then we were using a combination of field and lab methods to measure the volume and the sugar concentration of the nectar in all these plant species. Getting nectar out of a bunch of different flowers of all kinds of different shapes is actually pretty challenging. It's something that I worked on a lot as an undergraduate student, and recently have a preprint out about like how do you try to deal with those methods. So it was really cool to get to apply that in this project. And so once we had all of this, we then had the number of flowers and the amount of these floral resources in them. And that becomes the training data that I'm combining with the imagery. And so I have some very, very preliminary results. As I said, I got this imagery last week. But this is what an example of the predicted nectar might look like in a partial least squares regression model using that imagery. With the trees masked out. Of course, we don't know how many flowers are under the trees from aerial imagery. So we've had to ignore those. But this is as I said, very preliminary example, ton of work left to do here, but very exciting to get to see it take shape, several years after I kind of started thinking about this concept. So why did I do this? Coming back a little bit to kind of why did I do this? And I guess maybe the question also that was asked of me is a little bit of kind of what do I appreciate or what do I value about doing primary data collection as part of my PhD? The first piece is you get exactly the data that you want, of course, within some constraints in terms of time and funding. But when I heard about this project, it was like, wow, it would be so cool to have flower data that we can match up with this imagery, but that's not something that was being collected by this project at large. So it was up to me, if I wanted it, to go out and get it. And I was able to do that, which has been really incredible. The other thing is just you learn a lot about a system that is not captured in the actual data themselves. A colleague of mine, Jacqueline Fitzgerald, recently gave a really cool talk looking at kind of the information that is captured just in field notes that people are taking that is separate from kind of what ends up in the actual data sheet. But beyond that, also just like my ability to develop my intuition for what questions are interesting to ask, what hypotheses make sense, does my model output make sense to me? Does it seem to fit with what we're seeing in the field? All of those are things that I'm developing by spending time out there that I wouldn't necessarily have otherwise. And then you kind of have a similar thing in relation to your actual data. So understanding like what might be some limitations, what are applications that make sense or don't make sense, which outliers are things that we think might be real variation compared to errors, things like that. And then finally, just building a bunch of research skills. So right in this project, I was in the field managing a team of 5 or 6 people. I was writing a bunch of grants, writing these, again, proposals to request permissions, managing big research budgets, coordinating with all these groups in real time, right? All of those are skills that I want to be able to continue applying forward. And that's not to say that I think everyone here needs to go out and do fieldwork, but I did kind of want to end with a plug to, if at all possible, right, try to be talking to the people who were out collecting your data, if you're using data types for which that makes sense. And even if not, just talking to people who live and work in your system, right? Even though I've been working in this particular system for 6 years now, there's still a ton that I learn from people who spend a lot more time there or have much more long-term connections to those systems. Yeah, just want to end with huge, huge thanks to the folks who came out to help me collect this data. I absolutely could not have done this without them. And also to my funding sources. Thank you.
Okay, we're going to do another little costume swap here. Okay, so our last speaker this afternoon is Titilayo Tajuddin. Titilayo is a third-year PhD candidate here in CGA. She's co-advised by Dr. Katie Martin and Dr. Marcelo Ardone. She was a 2023-24 CCAS Global Change Research Fellow and a Mansour Doctoral Fellow. Before starting her doctoral program, she completed her MS in forestry here at State, using LiDAR data to estimate forest attributes. And she also completed a bachelor's degree in geography and planning from Lagos State University in Nigeria.
And then the radial retreat is going to be a circular movement of our line.
Is your mic on? I'm sorry to interrupt. It's sorry. People online can't hear.
Oh, my bad. So, and then each of these will represent our loss area. So in order to get the distance between the initial start step to the end step, that will mean the points at which our forest loss keep expanding. So that means we could see a center line, and then the line between the point to the edge will represent our retreat length. The same thing with the entire loss, and this is also going to show the linear retreat rates, like length, the transects, and then the radial retreat. So along each of these transects, we develop some kind of, we sample some points along each of these lines to get a spatially explicit retreat trace for this location. And in order to do that, in order to get the retreat rate, we calculate the length of each of the transects, and we divide it by the number of years in between the start year and the end year. So the start year could be 2010, and then the end year, the start year could be 2010, and the end year could be 2021. And this will be what our retreat rate map is going to look like for each of our conceptual retreat rate type. And some of the results for this analysis is showing what our classified map for 1985 is to 2022. And if you can see here, all of the black boundary or line is showing what the boundary of 1985 across all year, while the tree line for each year represents the red line. You can see that there is a very high shrub cover in year 2000, which some of those areas overlap with is burn area, like there was like some kind of wildfire in some of those areas in year 2000, as well as maybe there could also be drought in those periods. So we also observed that before our forest initially transitioned to marsh, it seems there is a kind of transition from forest to shrub, and ultimately some regrow, and then those that didn't regrow made it to being marsh. So that is about 82% of our forests that initially transitioned to marsh in 2022 has initially been classified as shrub in the year in between those periods. And then our linear retreat, that is horizontal shifts linearly, accounted for about 85% of our forest loss type or forest retreat type. Then our linear retreat rate, we observe that if we are looking at a longer time step, that is, there is a lot of year in between, the retreat rate is likely to be lower. So we have an average retreat rate of 4.2 millimeter year. That is, horizontally, our tree line is moving by 4.2 millimeter per year on the average. While there are some areas that are already experiencing about 53.5 millimeter retreat rate. So as you can see in some of these areas. But if you are comparing to a shorter time set between 2010 and 2011, because we observed that majority of our forest loss started post 2010, or more like 2010 onward. So we observed that for those areas, the retreat rate will be a lot more larger, just because we have a shorter year in between that we are dividing by our retreat transects. Then we also look at how different climate factors and sea level rise trend contribute to this retreat rate. And as you can see here, across all of our climate variables, we observe that there is an increasing trend from 1985 to 2022. So just showing that you could see the slope of this is for the minimum temperature is around 0.011 degrees Celsius. Same thing as precipitation, vapor pressure deficit for this area. Additionally, we also look at trend in salinity as well as sea level rise. One of the things that I wanted to highlight is that for the sea level rise, we observed that between our 1985 to 2022, there is an relative increase in sea level rise. That is, the relative sea level rise rate for this location is 5 millimeter per year compared to 1985 to 2010, which is 3.4 millimeters. So that is, we are seeing increasing rate of sea level rise in this environment. Then our model is showing that about 75% of the variation between our lateral retreat rate could be explained by all of our predictor variable. So which is very interesting. However, we could observe that there could be some underestimation of large retreat rates in some area, as well as overestimation of small retreat rates in some area. As you can see, the points that are above and below the red linear fate. Also, we observe that precipitation is one of the major predictor of our lateral retreat rate. So accounting for about 20% of our model prediction, followed by distance to coastline, because some of those areas that are much more closer to coastline are more likely to retreat faster compared to areas that are very far away from the coastline. Similar with high salinity as well as relative sea level rise. Elevation also is one of the top predictor of lateral forest retreat. Then in order to look at what this retreat rate is going to be like in the next few years or into the future has not happened. So there are so many forecasts or projections that is available, or not so many, but there are so many scenarios of things that could happen. It's not exactly what is going to happen, but at least looking into the future. And some of those socioeconomic pathways include SSP5, which indicate the worst case scenario. So in the event that every country, every economy decide that we are not going to embrace green economy, we just want to stick to fossil fuel development, so that would be the worst case scenario. And then the best case scenario is going to be everybody decide to say that, yeah, we are going to take the green road. We want everything green. So a very high sustainability, then that would be the best case scenario. In between is the middle of the road. So we all meet at the middle. So some people make this decision that we want to do this or not. At least there is a middle ground. So for this analysis, we are just going to be working with the SSP5, which is the worst case scenario, and the middle ground. So for the projected data in this environment, we observe that between 1980 to 2100, which is the year that we are thinking we are going to be projecting into, we observe that relative sea level rise would have increased to about 11 millimeter per year, which is very interesting compared to our 5 millimeter per year by 2100. Same thing, you could see increasing trend in minimum temperature, maximum temperature under the middle ground scenario. However, the worst case scenario, you could see it is around 13 millimeter per year, which is like more than double of our current sea level rise rate. Same thing as looking at minimum temperature precipitation. So these are just the data. So for all of our predictor variables, some of them don't have projected information. So we are just working with those that have projected information. But for the climate data, it is so coarse. It's about 28 kilometers square by 28 kilometers square. It is very interesting that there is a projected data, but that does not represent what we want, because some of the pixel for this location is just 30 meter by 30 meter. And using a 24 kilometer by 24 kilometer may not be a good representation of what we are looking for. So for now, I am still stuck with objective 3. I started already, but I'll keep working on that. So, but in the event that I'm able to get that done, I am interested in looking at what will the forest retreat rates be in 2030, 2050, and 2100? And where would the major forest retreats be occurring? So we could observe some of those areas that is having a very high retreat rate of about 53 millimeter per year. So would there be some other places? Where is our forest going to be retreating into in the next few years? And yeah, that's all. I'm sorry for people online for not hearing me.
So if we could have all 3 of our speakers come up, and if you have questions for any of them, you will have a chance now. Anybody? Yeah.
Great presentations, everyone. I have a question for you, Eli. I'm curious with the buckets of uncertainty. It's super cool that you guys are like basically able to inform where you can improve data collection, but I'm curious if there's any efforts to like make improvements to the underlying models themselves. Like if there's like a lot of uncertainty from one of the buckets, like if that is like a term in the model that is like subject for possible enhancements in the future, if that's been like a kind of like guiding model enhancements for POPs or for any of the other models, or if it's so variable between where the model is applying what the drivers of uncertainty is that it's hard to like make those like widespread improvements.
Yeah, that's a good question. I think a lot of that kind of questions about, are we making the right choice of model, often end up falling into like that process uncertainty bin, is where you'd kind of find model choice being more effective. But process for us also really includes a lot of our random draws. So that is definitely a way that you could look at it, is saying, if we feel like we're like well calibrated to our parameters, but our parameter uncertainty is still really coming out as like a dominant source, we could be saying maybe we're handling the parameters wrong. But yeah, I think it would be very challenging to determine why for something like that. Whereas yeah, yeah. I hope that answers it. Okay. Yeah.
Yes. Great presentations. Another quick question for you, Eli. How did you determine the cutoff for when one source would be dominant versus your gray pixels?
Yeah. Yeah, so it's just a simple like max. So basically we get like a percent number for contribution for each source for each cell for that year, and just pulling whatever the maximum was. So if they were all equal, we'd call them gray.
Exactly equal?
Yeah,
Or?
exactly equal. So yeah, that map is I don't want to say misleading, but just because you see a color, it's hard to know whether you're seeing that color because it's accounting for 60% in that cell or 26% in that cell. So those both show up as the same. But you could do a thing where like when we dove into that like subregion, you could make that subregion really small. Like if you had a smaller subregion, I picked actually a pretty large subregion. Um, yeah, let's go up, up. There we go. Oh, back down. One more down. There we go. Yeah, like I picked a pretty large one here. But like if you like picked like a real small one like that, you could make like the graph on the next slide for that one, and then that tells you like how much bigger we're looking at. Even though the like if you go to the one more slide, sorry. Yeah, even though like here it's like we can't tell from this particular graph whether like this green pixel here is green because that green's 60% or 25, not 25, 26%. We just know it's the biggest. Yeah.
How important do you think that is to know? Look at that.
I think, yeah, I think you'd want to use both of these like in combination with each other. So like if you were say interested in managing like a specific region, like if you were like a protected area, something like that. There's a protected area and you're interested in like within your boundary of your protected area. I think something like the graph for that protected area, like the stacked one, would probably be more important for you. Whereas like if you're looking at a wide area like this, I think it's kind of like that's why I was almost like not focusing as much on like the exact, there's a lot of like almost noise going on here, but just like this general pattern. And so I think they're both useful to use like with each other together. Yeah. Okay. We've got, okay.
Really, really great talk, everyone. My question is for Titilayo. So I was shocked when you showed that I guess the lowest rate of I'll say migration, but that was not the term that you used, was like 5 millimeters per year. What was and this is something maybe you mentioned, but I missed. What is your minimum unit area? Like what's the data that you're using as input so that you can get that 5 millimeters per year?
So it is actually in meters. I just
It's
bump it out.
meters, not millimeters.
So I was using 30 meter by 30 meter resolution data because I couldn't get millimeter data, but I just converted it to millimeter to make it a lot more easier to communicate. Because if that is going to be in meters, it's going to be like 0.0. So explaining that will not be able to show the variation across all of my study size. So it makes sense to represent it in millimeter. But the pixel size is 30 meter by 30 meter.
Okay, so you were not using like LiDAR.
Oh no, no,
Okay, that makes sense then.
no.
Thanks. I saw a hand around here, but I lost it.
Question for Eli. So you mentioned it on your slide there, the noise. So I was surprised there is so much variation. Do you have any like explanation or I don't know, something?
Yeah. Yeah, I think the explanation here is that, like we were talking about earlier, how you can't tell from this whether you're seeing like a 26 or a 60, is that in general, our numbers are a lot closer to 26s than 60s. And that's what's basically going on, is that for the majority of these cells, even the biggest source is not accounting for a majority of the uncertainty in that cell. Which is a useful result. It's kind of unfortunate in the scheme of management because it's not like there's a really easy answer and it's just like collect on host and you're done. But that I think what's making that noise happen is that a lot of these largest sources are like 30%, 28%, 35%. And so because we've got a lot of that, just that like cell to cell, yeah.
Have an online question. It's for Titi Leo. What DEM data did you use for elevation? How did you deal with the less than acres water body vertical errors?
Oh, so I use the cone DEM. It's majorly for the Eastern North Carolina, I guess. So it's around 1 meter resolution by 1 meter, but I scale it up to 30 meter by 30 meter to match up with my Landsat because I use Landsat imagery. Then for the elevation, so this study is supposed to show vertical retreat as well. So that is why I take it out, because elevation in this area, it is very hard to, because some of those areas has since changed due to erosion, deposition. So there are some areas that I'm seeing negative, and I think I cannot explain it to the audience. I would rather just not look at vertical retreat. So for the elevation, I just work with the coastal data, and every pixel that they classify as less than 0, we just bump up to 0, so that everything will just be 0, like flat. Yeah, I don't know if I answered the question very well.
Thank you. I don't know either. This is for Gwen. I applaud all the work you have done. So I kind of have two questions. One, for the program, do you see ways to integrate more data collection into coursework or internships or the degree program? And then the other is just, you got into it some, but how collecting the primary data is allowing you to facilitate conversations with communities and stakeholders.
Yeah, so trying to think about, so for first question about incorporating things into coursework, I mean, I do think that some of the pieces about particularly like understanding things about the data themselves and how to use them, I think a lot of that can be in some ways effectively communicated by really short-term things, by like going out and saying like, what does this method actually look like on the ground in like a day and a weekend, like that kind of structure can get you some of that benefit, even if you don't get the kind of like long-term exposure to that particular area in the same way that you would otherwise. That seems like the main kind of relationship to coursework. I mean, I think the hard thing about it is just all of the like, it takes a lot of time, it takes a lot of funding, it takes prioritizing fieldwork as part of what you're doing, and that requires support from your program, and it requires support from your advisor in order to get that done. And so I think there are things potentially on that angle in terms of the program that could make it easier if more people want to do that. But on a coursework side, I do think there is a benefit to doing even that short-term exposure to like the types of methods people have used to collect a data set that you might be using in terms of your understanding of how to use that data. Second question, how does it affect my interactions? In terms of the fieldwork that I did in Colorado, that is mostly like me in a field. So I learned a ton about that ecosystem itself, but that particular situation in many ways is really incredible because you're at a research station that is like, you are surrounded by all these other people doing fieldwork in this system. And like, I'm doing the fieldwork that I was describing, but there's other people working on insects and mammals and other aspects of plants and like all of this other work that's happening. But in general, you do get, I will say, a lot of kind of practice of your elevator pitch. Like, you do have a lot of people who are like stopping on the side of the road and being like, what are you doing? And you have to like be able to communicate that really clearly to them. But you weren't interacting with a kind of broader community of people living there as much in that system. The research that I'm gearing up to do this summer is sampling all over the city of Raleigh for floral resources again. And so that is already, I'm in the stage of like calling lots of businesses and being like, hi, flooring company, can I look at plants that are like the plantings around your business? And so there's going to be a lot more of like actually coordinating with people. Again, talking to them in that kind of elevator pitch way, like, why are we doing this? Why are you seeing these people in your community doing this data collection? But also getting more of a sense of like, in that you're going to get the kind of interactions where you're asking people like, why are they maybe making some choices, making different choices? Some of the outputs of that will go into things like extension documents in terms of people who might be making choices about their landscaping, all of that kind of thing. So that'll be built more into my next project. Any other questions on what?
Just a suggestion from Doug Newcomb for Titileo to try the DEM for Alligator River National Wildlife Refuge, and I'll share the link.
Okay. All right. Thank you.
I have one more question.
Yeah. When this is for you too. You show us preliminary maps with your nectar estimation. I would love to hear your thoughts on 1, 2, 3 different end users or stakeholders that would directly use those maps, that data.
Yeah, so in terms of end users in this particular system, there's going to be a lot of, it was a really great place to try to do this because there are a lot of end users to that data, in that there are a ton of people researching flowers and bees in that actual system. And so the coverage area of my ultimate, so I showed like one meadow, but the premise is to like make that map for that valley that I showed at the beginning. And there are like literally hundreds of bee research sites that are in that domain. And so that can potentially be kind of predictor data that could go into their research outputs. So in that kind of system, I'm mostly envisioning kind of that input. And then the other thing would be to do other analysis. And I'm also doing a little bit of that. So I was also that summer, I did not have time to get into it, but also collecting data about bee populations that I'm going to try to use to say, like, does this product improve our predictions compared to transects that I was also monitoring at the same time. But there are also some questions there about the distribution of plants and things that also become potentially useful for people. And then in terms of making a similar type of map in Raleigh, there's also all these questions about where do you plant things, where should we, like, where are areas where it would be beneficial to go out and add resources that would really improve things like the connectivity of the landscape for pollinators, all of those kinds of questions. Thank you.
Can we get another round of applause for our speakers? And thank you all. You are impressive and inspiring. And have a great afternoon.
I forgot to turn this on. I'm sorry. It's okay.
Once you put it back on yourself.